Thank you everybody for being with us today. We're going to get started, as usual, a few minutes late because, Miami time. A lot of you are new faces. I'm really glad to see you here today. My name is Ramya. I am the program manager for the Citizen Science project for the Key Biscayne Community Foundation. For those of you that don't know, we do monthly lectures on various science-related subjects. This obviously is one about Alzheimer's. We always send out flyers in the Islander. We post them online. If you want to be added to our mailing list for emails, just let me know and I'll take down your email and add you to our list. And with that, we'll go ahead and get started. By the way, our lectures are always the third Thursday of the month, unless there's some kind of holiday or something going on. That's you can usually expect that. So even if you don't hear about it or see anything about it, if you show up here on the third Thursday, you're probably in luck for a lecture. So with that, we'll get started. Today we have with us Dr. Nick Kron. He is a PhD graduate from University of Miami in computational biology. And currently he's working there as a postdoctoral associate and doing work with in the Aplysia lab and looking into how Aplysia, which are a type of sea snail or sea slug, how they can help us study Alzheimer's and other degenerative neuro conditions. So with that, I will go ahead and hand it off to you, Nick.
Thank you. Can everybody hear me in the back? Yep, okay, fantastic. So thank you, Ramya, and thank you to the Key Biscayne Community Foundation for inviting me to speak tonight. And thank you so much, all of you, for coming and listening to me. Tonight I want to tell you a little bit about invertebrates. These are animals without backbones, and how we can use them to illuminate things about human diseases like Alzheimer's disease. So before I start, I'm just going to give you a little bit of an overview of the talk this evening, just so we're all oriented. It's going to be in 3 parts. So in the first part, I'm going to discuss what Alzheimer's disease is and talk a little bit about its impacts in our society and the kind of research that has been done in studying Alzheimer's disease. Then I'll move on to talk a little bit about these invertebrates and what research has been done specifically in these invertebrates about Alzheimer's disease and what we've learned from them. And then the last part, I will go into Aplysia californica, which is my research organism of choice, and tell you a little bit about some of the recent research I've done in this subject area. And during each of these sections, there will be a banner at the top of the slide with the same color and with the word describing the section that we're in. So in case you zone out or whatever, you can always look up there just to orient a little bit. So, Alzheimer's disease. We hear about it all the time, but what is it? So Alzheimer's disease is the most common form of dementia. Now, dementia, we hear it a lot, in reference to people who are maybe starting to lose their memory a little bit, but it's got very specific clinical definitions. So clinically, a dementia is an impairment of memory and an impairment of one of any of the following other mental faculties. So these include speech, object recognition, so someone gives you an apple, you recognize that it's an apple. Motor activities, so this is, can you walk normally, can you use your hands normally? And then some more abstract things, abstract thinking, soundness of judgment, and complex tasks. So this is, thinking about what you're going to do in 3 weeks, whether or not, driving is a good idea, or completing a complex task like a Sudoku puzzle. Right? So if you experience impairments of memory and impairments in one of these other areas, then you're likely suffering from dementia. Now the important part is that it's not just, I get forgetful sometimes. It has to meaningfully impact your ability to carry out your day-to-day life. So if you can't drive to the grocery store, or if you forget the names of your loved ones, right? These are serious impacts, and that's when it starts to move into what would be clinically described as dementia. So Alzheimer's disease is the most common form of dementia. It's estimated that roughly 80% of dementia cases are Alzheimer's disease. Before I get into some of these statistics about Alzheimer's disease, what distinguishes it from other dementias, right? I mentioned there are a million different kinds. Alzheimer's is the most common. So Alzheimer's is distinguished from other forms of dementia because of specific protein pathologies. So proteins are the things that make up our bodies, and there are specific things wrong with specific proteins exclusively in Alzheimer's disease. These are considered diagnostic, if you will. So the first protein pathology that is heavily associated with Alzheimer's disease is what are called amyloid beta plaques. Now, this protein, amyloid beta, sometimes you'll hear it referred to as beta amyloid or just AB. It'll be abbreviated like this for the rest of the talk as AB. It is a protein that is important in the connections between your brain cells. So our neurons communicate with one another, and that's what, at a larger level, leads to your ability to listen to me and understand what I'm talking about. So these cells need to communicate, and this protein is involved in that communication. But in Alzheimer's disease, these proteins get damaged, and they begin to accumulate. So the body can't get rid of them, and they become sticky. They stick to each other, and they stick and stick and stick until they form this big sticky mass that we call plaque. So you can kind of think about like the plaque on your teeth when you don't brush your teeth. That exact kind of thing is forming on the outside of your brain cells when you have Alzheimer's disease. And that has some serious implications. Like I said, the amyloid beta protein is important in the connections between your brain cells, and when these plaques begin to form, it actually impairs the ability of your cells to communicate. And if a neuron can't talk to its neighbors, it eventually starts to wither and die. So these plaques actually damage your brain cells and eventually can lead to neuron death, which then gives rise to the memory loss or the other impairments that are associated with Alzheimer's. The other type of protein pathology that we see are called tau tangles. Tau is another type of protein. Tau works inside of your cells in this superstructure that you can think of as the railway of the cell or the highway of the cell. These are these long cables that your cell uses to transport important molecules, nutrients, proteins to and fro so that the cell can do its normal business. You can think of like a highway in a city, right? So a healthy tau is an important part of this. It allows your brain, the cells in your brain, to do their normal thing. However, in Alzheimer's disease, these proteins get modified, and they, like the beta amyloid, become sticky, and they stick to each other and eventually form these long threads. You can think of them almost like hairballs. And when these build up inside of your neurons, it actually blocks these railways, these highways inside of your cells. So it's like a hairball or a traffic jam inside of your brain cells. And this means that it's not getting the right nutrients in the right places, the right proteins to the right parts of the cell at the right time. And again, this leads to damage. The important thing is that both of these need to occur for it to be Alzheimer's disease. Some other forms of dementia may have one or the other, but when they both co-occur, that's diagnostic of Alzheimer's disease. And the key point is that amyloid beta alone is a problem. Tau tangles alone are a problem. But together, they have synergistic and multiplicative effects. So that is to say, having both would do more damage than we would expect of having just one or just the other. So they work together to make things even worse. Yeah, it's unfortunate. And it's likely that the damages come from the same place. And as far as we know, Alzheimer's is unique to human beings. So we do have evidence that some animals develop some of these types of plaques. We've seen it in dogs, we've seen it in some primates, but we've never observed it leading to a dementia in these animals. Now, it's not exactly clear why. It may be because of our complicated brains, but it has been postulated that it's actually related to how long we live. So they believe that it would be possible that these organisms, these dogs or primates, could develop Alzheimer's if they lived as long as we did. But most of them don't live past, dogs, 15, 20 years max. Monkeys, very similar. So, our anticipated average lifespan, 70 plus years. Very few animals in the animal kingdom live that long to experience the full accumulation of these protein plaques and tangles. So there's an important component of longevity to the development of Alzheimer's disease. So now that we know what Alzheimer's is on a clinical level, I want to tell you a little bit about why you hear so much buzz about Alzheimer's, why we talk about it so much, why it's on everyone's minds. So it's estimated, and this figure is actually from 2013, so it's even worse now, but it's estimated that 1 in 9 people over 65 develop Alzheimer's disease, and 1 in 3 over 85 develop Alzheimer's disease. Right? So we all of us, God willing, live a long, healthy life, but there's a good chance that any of us, as we age, will experience Alzheimer's disease, or someone we love may develop Alzheimer's disease. So it's very much something that most people will experience. It's also the 6th leading cause of death in the United States, and 5th for people over 65. So it's a big impact on the quality of life of many people in the United States. And as in the year 2012, there were roughly 17.5 billion care hours provided to people suffering from Alzheimer's disease, costing the families and the patients almost over $203 billion. So this is a lot. People are paying a lot of money to deal with this very terrible disease. And it's actually become more prevalent. Over the course between the years 2000 and 2010, it increased 68% in the United States. And it's actually projected to increase even more as the baby boomer generation, is getting older. They're, among the largest generations the United States has ever seen, and as they age up, it's becoming even more prevalent. And unlike a lot of other major killers in the United States, such as heart disease, the incidence of Alzheimer's disease has actually gone up as opposed to gone down. So there are many risk factors for Alzheimer's disease. Some of them are genetic. Certain genes, certain forms of certain genes lead to a predisposition towards the development of Alzheimer's disease. It's by no means a completely heritable disease. There are forms of familial Alzheimer's, but the most common form, the sporadic disease that most people will experience, has genetic influences but isn't absolutely determined by our genes. There's also a trend with traumatic brain injury. So if people who suffer from concussions or other kinds of traumatic brain injury are at increased risk. But a lot of them also have you could say, a social justice element to them, in that people with lower education are at greater risk of developing Alzheimer's disease. People with lower social engagement are at risk for Alzheimer's disease. So this is, I would say, kind of topical, considering the last year we've experienced with the pandemic and not being able to socialize as much as we'd like. That is an increased risk factor for the development of Alzheimer's disease. Heart disease, so not everyone can afford to eat well, exercise, eat the right things. So if you have heart disease, you're actually at increased risk of developing Alzheimer's disease. Part of it seems to be that whatever's circulating in the blood makes it to the brain and can trigger things. But the biggest factor, as I mentioned, is age. That's an inescapable truth of human life, is we're going to age, and that is the greatest risk factor for this disease. So something very much to consider.
What about medications? Certain medications?
I actually will get to that in just a second, no worries. So Alzheimer's disease was first described just over 100 years ago, but we didn't really start putting a lot of research effort into it until around 40 years ago, which is still a long time ago, but people have been thinking about cancer for a similar amount of time. And prior to 2011, the annual funding was just under $500 million that the U.S. government would appropriate for the scientific research of Alzheimer's disease. As of 2020, that has gone up to $3.1 billion allocated annually. This is a serious disease, and the government is taking it very seriously. And this is not including private funds and things like that may also fund research. So this is taxpayer money being put towards research to this disease. And in the year 2020, it was estimated to have cost the U.S. economy $305 billion in medical-related expenses. So it's, for people who maybe are a little more cold-hearted and aren't considering the emotional and social impacts, it's got serious economic impacts as well. And despite the massive impacts and how long we've been studying and how much money we've put into it, there have only been roughly 5 approved drugs for market. And unfortunately, these drugs only provide what clinicians would describe as modest clinical benefits. So they curb the worst elements of the disease, but they do not stop it, they do not reverse it. And I don't want to take away from that. It's great that these medications exist to help people who are suffering from this disorder, but one would think with all the effort and the amount of time that we're thinking about this disease and the money we're spending on research that maybe we'd get a little bit further. But it turns out that Alzheimer's disease is a very, very tough nut to crack. So there are a lot of difficulties with studying this disease. For one, it's very difficult to do research on people. And this is for good reason. You need to protect patients. You need to be considerate of their consent and their well-being. But this disease, it occurs in the brain, which means that it's very hard to access samples, right? I can't just walk into an office, get you to sign a form, and take a biopsy of your brain and study that, right? That would lobotomize you. That's frankly not possible, nor would I want to do something like that. So the only samples that we can get are from people who have donated their bodies to science after dying. And that, it's fantastic that people are willing to donate their bodies to science like that, and it gives us a window into this disease. But the problem is we're studying brains that have already succumbed to the disease, right? We don't learn anything about how it develops. Where does it start? We can't learn about that, right? So that's a huge impediment to studying the development of the disease on the cellular level. Another problem is, of course, like I mentioned, it's multifactorial. Your age, your diet, your ethnic background, the amount of social engagement, your education, how much metals you've been exposed to in your life. These are all playing parts into the development of this disease. So it becomes this difficult web of complex variables to disentangle. What's really causing what, and what comes first, chicken or the egg, right? That's very difficult. So we can't really study the development of this disease in people. We can look at other models. We can look at primates. We can see the primates that develop plaques that are similar to these that we see in Alzheimer's disease. Primates are just as difficult to work with ethically. These are thinking, feeling, living creatures. They also live a long time, so it takes a while to be able to get to a point where you can do the sort of sampling, and not everyone's raising monkeys, right? So monkeys are hard to work with. What's next? Mice. We've all seen lab rats, lab mice, very popular, an incredible model, very close enough to us that a vast majority of our primary medical research is done in mice or rats. They are fantastic, but they also are not a silver bullet. Their brains are just as complicated as ours are. Many of the same problems in studying the brain directly apply to mice just as much as they apply to people. So, and one of the major criticisms that has been levied against the use of mice is that they can only really replicate one element of the disease at a time. Frequently, when they're replicating the protein problems that we see in Alzheimer's, they're looking at mice that develop these in young age because they've been made into transgenic mice. So we see the protein pathology, but it's not in the context that the disease develops, right? They're either young, or they're young and they have the protein problem, or we're just looking at old mice. So they can't quite capture all of the elements of the disease, which makes it difficult to attribute the results to what might be applicable to Alzheimer's. And they are still feeling, breathing, thinking animals that have ethical concerns. You need to make sure that they are living in cages with sufficient stimulation. You want to make sure that these animals aren't suffering until you have to do the necessary part of sampling their brains. And there's also cost and space concerns, right? You have to have a room dedicated to raising mice. You need people to feed them and wash them and make sure they're eating the right thing and that they're healthy and give them antibiotics if they get sick. So there are costs associated even with mice. So that's why many researchers opt to turn towards invertebrate models for studying human diseases. So the first most popular model that I'm going to talk about is Drosophila melanogaster, aka the fruit fly. These are very popular. They've been used for a variety of research in biology. They're particularly great for genetic research because You can change only a few genes, and you can immediately see the change. Their eye color will change. They'll grow extra wings. They'll have fewer legs. Very popular model. Very powerful. And the other, probably most popular invertebrate model is this Caenorhabditis elegans, which, if you've heard of it before, you probably know it as C. elegans. But it's what's called a roundworm or a nematode. These are microscopic little worms that live. They're all over the place. They're on you. They're in you. They're all over the floor. They're in the air. They're everywhere. And they are also very popular models. For similar reasons, you can do a lot of genetic testing with these animals. And they're extremely popular in the study of longevity. So we've learned a lot about what we know about how aging affects the body through these worms. So why do we use invertebrate models when we have monkeys and mice and dogs and other animals that we could theoretically do this research in? Well, for one, they're very easy to grow. If you guys wanted to start a fruit fly colony, just leave a banana out, right? And then there you go, you're ready to go, ready to do science. Same thing with the nematodes, right? Like I said, they're everywhere. They're very easy to grow. You petri dish, a little bit of water, a little bit of nutrients, good to go. So they're very easy to culture. They also reproduce quickly. So you can create generations upon generations and generations of flies in a fraction of the time that you would with mice. So if you're looking at a disease that's associated with old age, it benefits us for having a model that gets old quickly, because we can look at old animals much faster than we would with a monkey that takes 10 years to get old. The important part is that they share the same fundamental makeup at the cellular level that we do. A neuron is a neuron is a neuron is a neuron. We're all animals. We all share some simple basic components. You can think of these as like the Lego bricks that we use to build up the brain. And while maybe the human brain is a big, beautiful castle made of Lego bricks, and a fly is maybe a little house, they're still made of the same Lego bricks. And Alzheimer's is, at its root, a disease in the neurons, in the little cells, those basic building blocks. And those are the same, or close enough. We have more types, but So fundamentally, it's the same system. So things that we learned about changes in a fly's neurons, we can usually assume it's going to be very close to what would happen in our brains under a similar scenario. And usually you test it in flies, then you test it in mice and see that if it is the same. Usually it is. And then you can start moving towards clinical research in humans. At the same time, their brains are less complex, and that's actually a benefit. Your brain is the most complex biological machine on the face of the earth. It's a miracle of nature. And that's actually very hard to study. How am I going to disentangle how thousands of cells are working together in this complex network, talking to each other, exchanging information with each other? It's nearly impossible to disentangle what each individual little cell is doing. But in a worm that has 300 of these, I can know exactly what each and every cell is saying, doing, growing, what's affecting the exact same cell every single time. And that's incredibly powerful if you're doing primary research looking at exactly what's going on at the cellular level. Associated with this are what we call reflex behaviors. So if you go to the doctor will ask you to hop up on the table. He'll get out his rubber mallet and he'll hit you in the knee and your foot pops out, right? That's a reflex behavior. You don't have to think about that. There's a circuit from your foot to your spine and back down to your foot that tells you what to do and when you get hit in the knee like that. These animals have conceptually very similar reflex behaviors. They vary from animal to animal. But a lot of the circuitry that's involved in that is much simpler because they have smaller brains. So it's easy to study not only what's going on with one cell, but a group of cells that determine a certain behavior. So if you're looking at a disease that involves changes in memory or behavior, like Alzheimer's, it helps a lot to have a simpler behavioral suite to work with. It's like learning how a bike works before learning how a car works. And then there's a lot of advanced genetic tinkering that we can do with these animals. You can breed flies to have all sorts of problems. They've bred flies that replicate heart disease just by breeding them. But you can also use tools like CRISPR, which has, I'm sure you've heard the buzzword CRISPR in the news. But we can change the genetic code of these animals to make them similar to humans. It's called humanizing. But we can make a fly express the human form of beta amyloid, so we can in a manner of speaking, we can give the fly Alzheimer's. And in these invertebrate models, these toolkits are very advanced, very efficient, and very cost effective. So we can test a lot of things quickly that will change the outcomes of whatever this analogous disease that we're giving these flies very quickly and very cheaply. There are few ethical concerns. There's always going to be a debate about the morality of inducing pain, but these very simple organisms do not have the same hardware that we do that generates sadness or experiences pain the way we experience pain. They also need fewer things. You don't need to enrich their lives as much. A fly is just as happy on a fresh banana as it is on a plant, right? So the ethical concerns are fewer, which is good on just a moral level, but also makes the paperwork a lot easier. So we can do more research faster. And they're cheaper, right? I can grow up a colony of flies and do a bunch of proteomics work in a fly for a fraction of the cost that it would take to do in mice or monkeys, and we can't even do this kind of stuff in humans. So these are all fantastic reasons for why researchers opt to use these invertebrate models over what we would call higher order models, mice, dogs, monkeys, because if you're trying to solve a problem, you want to have the best tool to solve that problem. If we're looking at just at what's going on at the cellular level, sometimes the mouse is not the best option. If you want to ship freight from one end of the country to the other, you're going to use a semi-truck, but you're not going to drive a semi-truck to the grocery store to get milk, right? There's a very specific set of things you're looking to address, and sometimes these invertebrates are much better options than a mouse would be. So I've told you that we can do a lot of research in these animals for cheap. It's easy, it's great, it's fantastic. But you might be thinking, well, I'm not a fly. What is a fly going to tell you about my brain? Surprisingly, a lot. So in reference to Alzheimer's disease specifically, we described how the proteins that are implicated in Alzheimer's disease work in a healthy brain, first in flies and in worms. Right? It's very difficult to identify these small moving parts in mammals. So we went to the fly because it's much easier to knock stuff out, tinker with things. And we found out that beta amyloid is important in the formation of the connections between our brain cells. It's also really important in responding to traumatic brain injury. It's part of the healing process, which is why you build some of that up whenever you have a concussion, puts you at greater risk for Alzheimer's disease. Same thing with tau. We know that it works as part of this cellular superhighway of nutrients and complex molecules. Now, because we discovered what it actually does in the cell when it's working as it's supposed to be, we also were able to tinker with, not me personally, but scientists, were able to tinker with how this beta amyloid works when it's mutated or dysfunctional the way it is in human Alzheimer's disease. And by observing that in these individual neurons, we learned how and why this beta amyloid is toxic. What components of the signaling machinery does it interfere with? So we found that out in flies and in worms. Also the role of metals. So, as we get older, we accumulate metals. That's just a fact of life. And these metals are important. Iron, aluminum, copper, these are micronutrients that your body needs. But if your body can't recycle them, get rid of them, use them appropriately, they build up, and then they can become toxic. And we didn't really know what role they played in Alzheimer's disease until we started looking in flies. We discovered that these ions actually push the beta amyloid into becoming this sticky, toxic mess, right? So again, we would have never found that if we were looking at rats, but because we had flies, now we know. Flies also help us see how the mutant beta amyloid plaques actually lead to memory impairment, right? It's one thing to say, okay, beta amyloid is toxic. It's another thing to say it's toxic in these ways, and that's why it affects our memory. In flies specifically, scientists described that it activates a stress-associated pathway, and this increase in stress in the brain actually pushes it towards neurodegeneration, right? The stuff that leads to brain illness. Similar stuff was done with tau, especially in the worms. So we learned about what tau does and how tau becomes toxic. There's a very specific set of genes that we know that modifies tau. And when they're too active, they lead to this modified tau that forms these fibrils, these tangles. And because we could make this mutant tau in these worms and grow them up in droves, we could rapidly test all sorts of different compounds that may or may not have therapeutic potential. And so now there's a bunch of potential drugs in the pipeline now being tested in rats and mice that may become future drugs that are even better than the ones we have now in solving Alzheimer's disease. And I could go on and on and on and on, but it would get pretty boring. But what I hope I've impressed upon you is that not only are they good alternatives to mice, they're even, in certain scenarios, better alternatives to mice. And a lot of what we know about this disease and about a whole host of other things that I didn't even go into, we learned in these very simple organisms. So these invertebrate models have wonderful translational value. They're a huge asset. I talked about the two big ones, the two big boys. But that's not my research organism of choice, and so I want to tell you a little bit about the animal that I work with and why I think it's such a great model for studying Alzheimer's disease. So this is Aplysia californica. It's known as a sea hare. So it's described as a marine gastropod mollusk. These are very fancy, unnecessary scientific words, but marine, it lives in the sea. It lives on the California coast in tide pools. Gastropod is just, it's got a foot and a stomach, and that's it. It's a very simple animal. And gastropods are things like land slugs, land snails, the beautiful nudibranch sea slugs that you see. These are all gastropods, right? Now, Aplysia is not really a snail because it doesn't have an external shell, even though sometimes we call it a snail. And it's not really a slug either because it does have a shell, it's just on the inside. It's got a very small shell on the inside. So they're kind of in the middle, so we call them sea hares because they kind of look like rabbits, right, with the little ears. They're very cute, very pretty too. So they're hermaphrodites, which, that's a fun fact, but also for animals that have separate sexes, sometimes the differences in the sexes aren't accounted for in a study, right? It's sometimes difficult to account for what happens in a female versus a male. And this is actually a huge problem in the medical field, right? Because we know a lot about, how these diseases like Alzheimer's affect the male brain because we use male rats, because scientists are prejudiced and afraid to use female mice because, history. But so this can be a problem that you have to account for, and it's very important to account for this when you're doing studies. We don't need to worry about that. All our animals are both all the time, so it makes it even easier to study the simple cellular level effects. Like I said, from the California coast, so they're not native here, but and they only live a year, so that was great. For me as a PhD student, I had 5 years to get in, get out. I can grow up, 3 or 4 cohorts of these and study them in old age. So that's actually a really great boon for studying a disorder that's age-associated like Alzheimer's. And they are reared in captivity. So this is a big thing. If you're going to have a model organism, you're going to need to be able to grow it. Flies and nematodes are pretty easy. Slugs were a little bit harder, but thankfully we grow them locally here. So the University of Miami Rosenstiel School is actually the national resource for Aplysia. So we grow them, we ship them in from California, we breed them, and then those first generation progeny are used in research across the country and even across the world, right? We ship them to Israel and Japan and all sorts of places. So they're available locally, and the fact that we grow them here means that researchers like me have access to them from egg all the way to fully grown. So I can know absolutely everything about this animal, everything it's ever eaten, how big it's gotten, who its friends were, all of it. Just kidding, they don't have friends. They are very social though, they like to hang out together. So it's great, we have them here, and they're very accessible to anyone in the world if they want one. So what's so great about Aplysia? Well, a few things. For one, it's got like the other models, it's got a simple brain. There's only 20,000 neurons to keep track of. And we know more or less what most of them do, who they talk to, what kind of signals they send to each other, how many there are, what behaviors they're associated with. And so they don't quite have a brain per se. They have this group of what we call ganglia. You can think of those as like mini brains. They're kind of like, our brain has a frontal lobe and an entorhinal cortex and these regions of the body, or regions of the brain. That's what these are. These are regions of the brain. And they've got these big, beautiful orange neurons. They're really pretty under the microscope. So R2 is, one of the bigger ones. In fact, they are this big. They are so big that you don't need a microscope to see some of them. So this is R2, that big neuron I was just talking about, and this is a penny. So that's Liberty written on a penny. It's as big as the B on the penny. So you can see these. A human neuron is vanishingly small. You need an extremely expensive, big microscope to see it. Not with these guys. We can use much, much cheaper, much smaller microscopes, and we can look at them. We can poke into them with electrodes to listen to what they're saying to each other. We can know exactly that every time we're looking at R2, every time. Same brain cell in every animal. That's incredibly powerful. You can't do that with human brain cells, right? And then they have these same simple reflexes. So the most famous one with aplesia is called the siphon gill withdrawal, but essentially they have these beautiful parapodial wings, and in between them they have their gill. This is what they breathe with, and if you tickle their gill, they don't like that and they suck it in, right? So that's a very, very simple reflex. I'm sure if I tickled your lungs, you'd probably suck them in too. But the animal can learn that if you tickle its lungs, it's not going to get hurt, and eventually it won't freak out as much every time you tickle its lungs. So this is simple learning, right? And these reflexes are very simple. In fact, so simple that we know exactly the neurons that do them. And in the case of this one, there's only 3 or 4. So we can listen to each and every one of these neurons and know exactly what they're doing when we tickle the lung. That's incredibly powerful. I could never do that with a human being. I couldn't find the exact one cell that is associated with the smell of dandelions, right? Like you can't do that. But with these guys, you know exactly what cells are doing what when you're interacting with them. So I can know exactly when things start to go wrong and why it's leading to changes in these behaviors. That's incredibly important. And they have a very long history of research, actually. A lot of us haven't heard of them, but people have been working on these since, Aristotle was the first one to describe them, but at least since the 1950s. This is one of the reflex behaviors that we do in the lab that I work in. So that's me. This is an aplesia. This is called the riding reflex. So we drop them from the top of the water, let them sink down, and they don't like to be on their side, so they slowly turn upright. So it's a riding reflex, and they'll do this every time. Every time you throw them down into the water, they'll flip up, and they'll start to move. And we can measure how long it takes them to do this, and it takes them longer and longer as they get older, right? So their behaviors age just like ours do. So this is one of the ways we keep a kind of a bead on how their brains are doing, right? So a lot of my research is aging related, so that's the kind of stuff we keep track of. How much are they slowing down? They slow down just like we do. And so again, you may be thinking, okay, it's a slug. What does a slug have to do with me? Well, in the neuroscience field, there's a very famous researcher, Dr. Eric Kandel, and he won. He shared in the 2000 Nobel Prize in Physiology or Medicine, one of 3 who were laureates that year in this category. And he used aplesia as his model organism. What could have been so earth-shaking that in a slug that warranted a Nobel Prize? Well, he was interested in learning, specifically the most simple kinds of learning that exist in most animals. They definitely exist in aplesia. And he wanted to know, where do we store memories? How do we store memories? Because up to that point, we had no idea. So he was looking at the structures that encode our memories. And he found them in aplesia. That reflex circuit I showed you, those 3 neurons, he looked at how they interacted together as the animal learned and showed that it was the increase in the number of connections between these neurons that stored this memory. That's huge. He also learned the chemicals that these neurons were sending to each other as signals that led to the formation of these memories. That simple mechanism is in you, is in me, is in everything with the brain. This exact paradigm exists in all of us. So we know how we learn because of how we know that the slug learns. So the very basic fundamental neural circuits that exist in these very simple animals, they're in our brains too, and by studying the slug, we learn a little bit about ourselves. So I just want to show you why I think Aplysia may be better than the flies or the worms for Alzheimer's specifically. So here at the top, we have all our favorite models. We've got Aplysia here on the left. We got fruit flies or worms, our mice, and then that's me. Please don't experiment on me without my consent. So lifespan. Like I said, Aplysia live about a year. That's 4 times as long as the fly and a lot longer than the worms. But it's shorter than the mouse, and it's hopefully I live to be that old. And it's a lot shorter than our lifespans, right? And while it is longer than the fly and it is a lot longer than the worm, I think in this scenario this is actually a good thing. It's short enough that I don't have to wait for a mouse to be 3 years old to look at aging research, but it's long enough that I'm not going to miss anything. If this thing lives 3 months, if I miss it by a day or two, I could miss an entire important stage in their aging process. With Aplysia, I've got 12 months to work with, right? We've actually nailed it down to a T. We know exactly how these brains age. So I think for that reason, the kind of in the middle lifespan is actually very good. As far as neurons, 20,000, like I said, it's a nice tractable number. It is actually a lot less than the fly, which I found kind of surprising, but these flies have a little bit more going on up top than our slugs do. It's a lot more than the C. elegans, which is 300. Can you imagine operating your body with 300 cells? But mice have 70 million, and we have more than 85 billion neurons. How are we ever going to know what each and every one of those does? It's crazy. I think there's more cells in your brain than there is stars in the night sky. It's wild. So it's definitely more than this 300, but I think 300 is like maybe too simple, right? That's 300 in the entire body. It's not just regulating how this animal's acting, it's also regulating how it's digesting, things like that. So I think this 20,000 is a great number. We know there's enough of them that it's complex enough that we can say, okay, this is a brain and it's doing things that brains do, but it's also not so high that we don't know what each individual neuron is doing. And then the molecular toolkit that I mentioned earlier. So it's really robust in flies and in worms, and it's actually very good in mice too. We can create transgenics and all sorts of things very effectively. Doesn't really exist in us. There's been some controversial use of CRISPR in people, but as of now, there's no gene editing or anything like that in people, and that's probably a good thing. And we do have some of these technologies with Aplysia. It's not quite as advanced as with the fly, but it's enough that we can do the kind of Alzheimer's research that we need to do. And the big point that I want to make is that you'll see up here that we have, so this is what's called a dendrogram or a tree graph. And this is showing how related to each other these different groups of animals are. And the longer the branch, the more they've changed with time. And you'll see up here that the fly and the worm, they're kind of close together. They're both in a group of animals together. And they're pretty down the graph, right? They've changed a lot since the last common ancestor. But Aplysia is right here. And dogs, which are the representative for mammals, which is the group that includes us, they're on almost the same level. So Aplysia are actually genetically less distant to us than the flies or the worms. So conceivably, their genetic makeup is more similar to ours and can give us even more insight than we got from the fly and the worm. And as I showed, we've gotten a lot out of the fly and the worm. So that's very encouraging. So there has been some Alzheimer's research in Aplysia, not as much as I would like, but it was shown in 2005 that you can induce what is believed to be a precursor to Alzheimer's disease with the native genes inside the Aplysia neurons, right? So Zhang et al., what they did was they just overexpressed the gene that's normally in there, and they caused what are called cofilin aggregates, which are like a structure onto which the beta amyloid snaps onto, and then creates these plaques, right? So we can create these in Aplysia neurons, and we know the process by which it occurs. We can also, it's called transfect, we can inject these neurons with the mutant human tau and cause the tau pathology, these tau tangles that we see in Alzheimer's disease in these neurons. And they do very similar things. So Shemesh and Spira in 2010 and 2011, they actually created neurons with the mutant human tau in them. It gave them an Alzheimer's-like condition in these neurons. And then they started testing drugs on them, and they found a few that actually helped alleviate the tau. That's, it's got to work its way through the chain of, next tested in mice, et cetera, but there may actually be a drug in the future that actually came through Aplysia research, which is pretty exciting. And we also, in 1996, a while ago, we discovered how amyloid beta impairs neuronal function in respect to a specific small element of the way cells communicate in Aplysia neurons. So we've as Aplysia researchers, we've made our mark on this field. So my research was actually just recently published this week. And so what I did in the study here is I looked at it from a genetics perspective. So I'm a computational biologist. I look at genes. Genes are the blueprints for how we build our body. And I looked for genes that we share with Aplysia, right? So I showed you that tree graph, and we're, more so than the fly and the worm, we're pretty related to these slugs, and we share a lot of genetic material. So there are gene relatives in the slugs that are also in us. And I look for ones that are known to play important roles in Alzheimer's just by, people will sequence the samples from those people who have donated their brains to science. And we know that they're associated with Alzheimer's, but we don't really know what they're doing. So I was looking for those in Aplysia. And then I also looked at what's called RNA. So we know that DNA, it's what our genes are made of, and these are the blueprints. And RNAs are like the photocopies of those blueprints that you send out to the factory to build the proteins. So I can know what you need based on how many orders you're putting in. So let's say at a restaurant, which item on the menu is most popular. Let's say it's a hot dog, you're gonna have most of your orders are gonna be hot dogs, right? Same concept here. I know which genes are being copied the most, and that tells me what the cell needs or what the cell is experiencing. So I looked at that. So I identified 413 human genes that have relatives in Aplysia. It's closer to 900 in Aplysia, but so there's a whole lot of genes that we know is important in Alzheimer's disease that is also going on in these brain cells. Right, that's the 413. And importantly, Aplysia has native equivalents to the precursor to amyloid beta, the machinery that produces amyloid beta, and tau. So in studying these in worms and flies is how we learned about how they work in us, and Aplysia has these same relatives. So this means that we can do similar kinds of research in Aplysia. So that's encouraging. So then with the RNA, I was comparing the signatures that we see in these molecules, these copies of RNA, in Aplysia brains as they age with human brains that are experiencing Alzheimer's disease. And both the Aplysia, the old Aplysia neurons, and the Alzheimer's brains showed signatures of impairments in learning and memory, which you would expect. Mitochondrial dysfunction. So mitochondria is the powerhouse of the cell. It's what provides the fuel for the very expensive organ in our skulls. A quarter of your energy, of your food energy, is used by the brain. It's very expensive. And in Alzheimer's disease, these are usually experiencing dysfunctions. They're not working properly. And they're not working properly in a similar way in Aplysia. And then just like with Alzheimer's disease, Aplysia also experienced protein folding issues with age. They also showed similar increases in what's called neuroinflammation. So when your brain is sick, it swells. Same thing's happening in these slugs. Cellular stress, so all these protein aggregates are stressing your cells out. That's very damaging. And Aplysia neurons are experiencing something very similar. And also iron accumulation. So the same heavy metal stuff that I was talking about earlier, the heavy metals make beta amyloid more toxic. Aplysia neurons are also accumulating heavy metals with age. So we can study that process in Aplysia. Now I don't want you to get the picture that Aplysia are getting Alzheimer's. They're not. But they have the preconditions that are similar to the preconditions for developing Alzheimer's. So this research suggests that we can induce Alzheimer's in these slugs or something like Alzheimer's and use that as a means of studying Alzheimer's in a primary sort of way to point us in the right direction so that we use our money in the rat studies more effectively. So with that, I'd just like to quickly acknowledge our funding. It comes from the National Institutes of Health. It's a grant that's been running for like 15 years now. It's really fantastic. My PI, my boss, Lynn Fieber, she runs the Aplysia lab out at Rasmus, and Mike Schmally, he's the PI on the grant. And then, of course, the National Resource where we get all our animals, and the University of Miami Computing Center, which helps me do a lot of my computing work. And with that, thank you for listening, and I'll take any questions.
You said that we live longer than most animals.
Yes.
We develop. Okay. There are some animals that live longer than we do.
Mm-hmm.
Has somebody discovered Alzheimer's in, say, tortoises or something, the giant tortoise?
Oh, that's interesting. I'm not sure anyone has actually looked at tortoises in terms of neurodegenerative diseases, to be honest. That would be actually very interesting to look
There
at.
are animals who live longer than we do.
So one long-lived animal that they love to look at is the naked mole rat. So it lives 20 times as long as the nearest closest relative. And they're really interesting because they don't get cancer. That's usually what they're looking at. They're not really looking at their brains as much. But that's interesting. That's kind of a gap. Not a lot of people are looking at very long-lived animals for neurocognitive disorders.
Right. You can find dead ones every now and then.
Sure.
They don't die very often.
Yeah.
You could at least find them.
Yeah, that or elephants.
Second question,
Yeah.
too. What I remember at the very beginnings of Alzheimer's publicity, before you were born, probably, and was that people seem to think that aluminum was the culprit.
Sure.
And you mentioned iron. Is aluminum still
Yes.
implicated in the plaque?
It's not quite as central as maybe it was originally thought. It was thought that maybe aluminum was the source, and it's not the only cause of Alzheimer's, but definitely any of these heavy metals, so iron, aluminum, copper, lead, they get stuck in the brain and they can interact with the molecules that make up your brain cells and damage the proteins, and among those are beta amyloid, tau, right? So definitely if you have a high aluminum intake and your brain isn't getting rid of that aluminum effectively, that does contribute towards Alzheimer's disease. So
people were really worried about deodorants because
right, yeah. So that that's still an active line of research actually. Yeah.
Okay, so we know there are places where you can live and you live longer. Are there places where you can live where there is no Alzheimer's?
So there are communities they're called centenarians, people live over 100 years, that are of intense interest. They're scattered throughout the world. There's some in Okinawa. There's a one in Sardinia that's under a lot of research currently. And they look at the fact that a lot of them not only are old, but they don't have high incidence of dementia. And the research that's coming out of that area of the world is suggesting that there's a multiplicity of factors that go into why they're living so long, but they are a low-stress life, a lot of social interaction, lots of walking, and a healthy, balanced diet. And these are all things that individually have been looked at clinically with not only Alzheimer's, but dementias in general. And all of these are effective at either slowing the development or mitigating the negative consequences of Alzheimer's disease. So, decrease your stress, eat well, walk a lot, and make sure to, hang out with your friends.
What was the last factor you said on those originally?
So it's healthy diet, low stress life, lots of social interaction, lots of walking. Yes.
The last, and by the way, horses don't really have Alzheimer's. My daughter is a equine vet.
No.
So I asked her that question this morning. I was
Yeah.
curious. I was curious, it seems like in the last 10 years, there's been such an emphasis of over-the-counter vitamins. And people seem to take them without really consulting their own doctors. They just hear that taking iron is supposed to be good. The new thing with COVID was take zinc because it's supposed to be good. How does this affect Alzheimer's? Is it effective in any way?
So the tricky thing with supplements is that they don't have to go through the FDA. So they don't have to go through the FDA, right? So if you're taking a vitamin, they don't need to necessarily clinically prove what they say it's going to do for you,
What's in it?
or what's in it. right? So a lot of these are people will be extrapolating from a study. So they'll do a study in a rat neuron or a fly neuron, and they'll test a compound, something from turmeric, for example, and they'll say, oh, okay, this helps slow down Alzheimer's disease in this one cell, in this exact set of conditions. And they'll say, oh, the key is just eat a tub of turmeric a day and you'll never get Alzheimer's. It's not really how it works, right? They haven't considered how long it takes to digest, what it does when it actually enters your body, right? So a lot of these, I don't want to say that don't take your vitamins, but if there was a clinically testable vitamin that did work, we'd probably know about it at this point with the amount of scrutiny that's been gone into it. So while maybe vitamins don't work, again, the diet is very important. Eliminating certain foods, eating nuts and berries tend to be kind of associated with decreased risk, but we're not positive what it is in these things. We think it's the antioxidants, but it's hard to say, right? It's hard to study in humans. So does that answer your question?
Yeah.
Okay, thanks.
I read that there's a bad relationship between taking sleeping pills and in particular the counter kind that are antihistamines.
Oh, interesting. I didn't know that.
Yeah, I've read it in different health publications.
I believe you. What I do know is that what's called the circadian rhythm. So your body has an internal clock, right? And when you disrupt the normal sleeping pattern of the body with medications, it can have dangerous consequences for your brain cells, right? It stresses your cells out. And anything that adds on to the stress in your brain can push it closer to this threshold of developing Alzheimer's, right? So, you want to get a healthy night's sleep.
About the accumulating of heavy metals in the body, is there, let's say, a routine testing when to go for a checkup for your lab test? Are they looking at well, I know they're looking at iron and some other metals, but is there like an interest in identifying the accumulation of metals in those tests?
Yeah, absolutely. So I don't know if this is a test that you would routinely get while you're at the doctor, but it's becoming increasingly understood that the level of iron in your blood and the level of iron that's accumulating in your brain are maybe somehow connected. So increasingly, doctors that are looking at patients who have maybe are at risk of developing Alzheimer's, they'll keep an eye on that sort of thing. So these sorts of understandings are becoming more commonly implemented for specialists who are actually looking at these neurodegenerative disorders. But still early days.
Well, isn't chelation supposed to eliminate those metals?
It's hard to get the chelator into your brain.
Oh, all right. So,
And these metals are important, right?
it doesn't work.
Not necessarily. You can't just inject a chelator into your blood and it works, unfortunately. I wish it was that simple. Yes?
If you use your mind a lot, for example, if you study or you keep working longer, do any of those things prevent Alzheimer's or delays Alzheimer's?
Yes.
Not have any-
Absolutely, absolutely. So I mentioned a little bit earlier, but it seems that a risk factor is education level. So if you're less educated, you're more likely to develop Alzheimer's, right? So people who use their brain a lot, who keep training their brain, who will do Sudoku or work or crossword puzzles and things like that, and especially social interactions. So social interaction is actually very taxing on the brain, right? You're thinking about what the other person's doing, you're reacting to what they're saying. It's a very good brain exercise. And people who exercise their brains in this way either stave off or lessen the impact of the disease. So absolutely, if you're training your brain, you're working towards slowing down the development if you're predisposed.
Because I see people, for example, a doctor who's been really successful his whole life uses his mind a lot. And then when he retired, he developed
Yeah.
dementia. He stopped, all he did was read newspapers. He did do that, but other than that, he didn't do anything. And he did develop dementia after about 10 years.
Right, that's actually that's incredibly common, and it's something similar that's frequently noted is when a loved one dies, especially a spouse, people will, in their grief, will retract from the social life and then very rapidly develop dementia, right? So like you're saying, it's when you stop training that brain is when you give it an opportunity to creep in. So absolutely. Yes.
But in the example of, say, Ronald Reagan and Margaret Thatcher, who were presidents, prime ministers,
Sure.
into older years, they were quite aged, they developed dementia, Alzheimer's later in life, highly educated, very social, very active. That example doesn't follow.
So it's not a panacea, for one, but also you have to recognize that these people not only lived very active lives, they also led very stressful lives. And stress is a huge contributor to all sorts of disorders, diseases, heart disease, neurocognitive disorders. I mentioned earlier neuroinflammation, so that's increasingly becoming very much understood as an important factor in many neurocognitive disorders, especially dementias, is that the more stressed out you are, the more inflamed your brain is, the more it actually ends up producing misfolded proteins that it can't address, or it doesn't cycle its iron properly. So while social interaction is important, stress is also very important.
Your genetic makeup has something to do with it too.
Yes, so there definitely is a heritable component to Alzheimer's. There's last question. There's a gene called apolipoprotein, which is involved with, it carries fat molecules in the blood, right? And it's associated with a lot of disorders, heart disease especially, but it's also very much a risk factor for Alzheimer's disease. So if you have a specific version of this gene of apolipoprotein 4, you have a factor increase in your risk for developing Alzheimer's. And so this is something they can test. They can say, oh, you are at risk because you have this gene mutation, or you have a family history of Alzheimer's disease. Therefore, you're at risk. So absolutely, there's a heritable component.
There are several medications that have been promoted as not curing, slowing down Alzheimer's. What is your information about that?
So I'm not a medical doctor, so definitely don't take medical advice from me. Definitely consult your physician. As far as I understand, there are approved drugs that help alleviate the symptoms or the progression of the disease. I don't know too much about how they work or how effective they are. That's definitely a question for your physician. But some drugs do exist, right? They have approved some drugs. But I don't know if we've found a cure. That we haven't found yet.
What about the effects of alcohol?
It's not good for your brain. It's not good for your brain. They say drinking a little bit of wine is once a week or every day or whatever is healthy for you, but they flip back and forth on it. And there's a concept called hormesis where they say a little bit of damage is good because it makes your body work to fix it, and therefore you're warmed up to fix things, right? So that's how exercise works, right? You tear your muscles a little bit when you exercise, and then they come back stronger. So conceivably, drinking a certain amount of alcohol may contribute to that. But if you're consuming a lot of alcohol, it poisons your brain. Alcohol is a poison, and high alcohol use is also correlated with a lot of these cognitive disorders, heart disease as well. So don't drink too much.
So then what about coffee?
I don't know about coffee. I know there's a lot of interest.
A number of years ago, and then I read recently, and a couple of years or so ago, that coffee's good. So then I decided to drink it a couple of days of the week.
Yeah.
And I drink my herbal tea.
There's a lot of interest in caffeine. Caffeine, it affects your brain. That's why it wakes you up. But I'm always skeptical of these sorts of these dietary fads, if you will. Eggs are bad for you. Eggs are good for you. Fat is good for you. Fat is bad, right? As our understanding grows, our perspective changes. I like coffee. I drink coffee, but whether or not it's gonna, keep me sharp in my old age, I don't know. I will admit that I eat oatmeal with nuts and blueberries in the morning specifically to, keep me aligned. There are some studies that have people eat blueberries and not eat blueberries, and the people who eat blueberries seem to do better. And maybe it's the antioxidants, maybe it's this, maybe it's that. They're good for you. Don't eat too many of them. Consult a dietitian to make sure you have them in the right balanced proportion in your diet. But they're definitely a good thing to eat, I think. I do it, but again, I'm not a doctor. Consult your dietitian, your physician. Yeah.
Earlier that studying the aplesia is helpful in that they're hermaphrodites, so you're not running into that problem of studying one sex more than the other and creating that kind of bias. Is there any concern that you're missing a bias that should actually be there, that women are more affected in some way than men or vice versa, that you're not seeing because you're actually studying hermaphrodites.
Right. Absolutely. So in some ways, the fact that they're hermaphrodites means that we are unintentionally, just because of the biology of the animal, we're incurring the same research sin as the people who only use male rats, right? We're only looking at one sex. But at the same time, aplysia are both sexes at the same time. So, you know, they're cycling with all the same hormones as both sexes. So whether or not that's going to address what's going on in the female brain versus the male brain, I don't think the research that we're doing in aplysia is that far along in the research chain for that to matter that much, right? We're just looking at the cells, and your sex at your individual neuron level is not really that big of a deal.
Are there more men or women?
Alzheimer's
from
is actually more common in women. As far as I understand, it's more common in women.
By a wide margin, or just a little bit, or?
You know, I would have to go back and check. I don't want to give you a wrong number, but I think if memory serves, I think it's 2/3 are women.
Of course, they live longer too.
Right, they do live longer.
Mixed up factors.
Right, again, it's a very complex disease, right? But again, I'm not 100% sure on that figure. I'd have to go back and check. But as far as I understand it, it's more common in women.
Is the increase in numbers of people who are contracting or showing signs of dementia, is that just because there's more recognition in communities of what dementia is, and it has always been there? It's just that we didn't talk about it so much. You know, if we had a grandmother or an uncle or an aunt who was a little bit off, we didn't say.
Sure. Yeah, no, I think that's a fantastic point. It's like that's a question in any sort of scenario where suddenly you're seeing more of something. Is it really that there's more of it, or are we just suddenly looking now? And I think there is a component to that, but I think in parallel to that, you know, obviously, you know, it's only in the last 40 years that we're really seriously looking at this, right? So there's undoubtedly underdiagnosis historically, but also life expectancy in the last 100 years has skyrocketed, right? And the number of people and the number of people living to those ages has also grown. So part of the reason that we're actually seeing, as far as I understand it, that we're seeing an increased incidence of Alzheimer's in the United States is because the baby boomers were such a large generation. And now they're starting to enter that period where they're at increased risk of Alzheimer's. There may be other factors, you know, our lifestyles, things like that. But so I think definitely there was historical underreporting, but I think also there's unique demographic changes that are influencing this increased incidence, if you will.
And environmental?
For sure, absolutely. The amount of stress, what we're eating, how much exercise we're getting, all of that. I think life on planet Earth has changed so much in the last 100 years that I think there's absolutely a multiplicity of factors that's leading to this. So I'm looking at a lot of things. Right now I'm working kind of in parallel, something that may be partially associated with this. So these animals also experience a brain infection, a natural virus that infects their brains, which sounds like it's something out of a sci-fi movie, but it's actually more common than you'd realize. So I'm currently looking into that, and while it's not directly Alzheimer's, part of the reason why we're interested in this brain virus is because there's a theory of Alzheimer's that's been gaining traction recently that suggests that maybe it's not the exact definitive cause, but Alzheimer's disease has been known to be associated with brain infection. In fact, most of the people who are suffering from Alzheimer's disease have the genetic code of herpes virus in their brains. Herpes virus and cytomegalovirus. So this is a virus that's in all of our bodies. We've probably had it once in our lives. It's herpes simplex 1. But it's in our brains. When it gets into our brains and creates a secondary infection, it's known to contribute towards the development of Alzheimer's disease. How, why, is still under research. There's evidence to suggest that this beta amyloid, these plaques, occur because the beta amyloid attacks the virus to neutralize it, but then builds up too much of it and the brain can't get rid of it because we've gotten older and our machinery doesn't work well, and it forms these little nodules that eventually grow into these plaques. So there's some suggestion that there's a viral component to Alzheimer's disease. So we've got a brain virus in these slugs that happens naturally. We're hopeful that by understanding how that works and how the immune system of the neurons in our slugs attacks this brain virus, we may be able to understand how these neural infections contribute towards the development of Alzheimer's. So it's analogous, kind of on the side. But after this, I'm hoping to go to Spain. I wrote an application for a fellowship in Spain to look at something completely different. But yeah, yeah. But we'll see. There's a good chance I'll come back to this because there's a lot of good funding and it's meaningful work. So.
Before we take off, Sally, did you want to talk about the fidget blankets?
Yeah, I do. Okay, thank you. Boy, you are brilliant, and that was really interesting. This started a couple years ago here at the Key Biscayne Community Center. Ann Tainter was involved in this, and we made all these blankets. We made about 500 of them, and the blankets were originally these, and they are to be put on a table, and usually with a caregiver, and then the Alzheimer patient feels and touches. It's supposed to be very calming to an Alzheimer patient. If you've been around somebody like my dad, he got very agitated. And so if he had something to squeeze, it helped him a lot. Then the second thing we did was go into aprons, because you put this on an Alzheimer's patient and they don't lose it because it's always with them. These they'll leave or they'll give to someone. Cliff went with me one time and the two ladies got in a fight because they wanted this one and then the other lady wanted hers, and so a lot of children at that age, And then the last thing we are doing, we need knitters and crocheters, because this arm thing will stay with us, and just play with one of these things, touch it, feel it. And it always is with them. So we have people doing this. Knitting, if you knit at home, please bring us, knit at home, bring them in. Or crocheting. So, I'm always trying to sell this project. These are free to give out to anybody that wants them. We do accept donations. And when I turned 80 over in Fort Myers where we lived, I put a little pot out there and $500 was donated to me. So I'm out of that money now. But anyways, enjoy looking at them. If you have anybody you know, or you know somebody that works at a care place, please let me know, because I'd like to give them all out. Thank you.